Okay, everybody, welcome to our 14th episode of our WBESS series, Western Biology Evolution Seminar Series. For today, I have Dr. David Salamanca-Diaz from the University of Exeter in the Living Systems Institute. And he will be talking to us about the origins of coloniality, something that I find very fascinating and something that has been keeping evolutionary biologists busy for decades, if not centuries now. So, David, the floor is yours, and yeah, take it away. I look forward to it.
Perfect. Thank you, Mateus, for the opportunity and the chance to present my work. Um, yes, my name is David Salamanca. I am a postdoctoral researcher in the University of Exeter in the lab of Jordi Solana. I've been here for 2 years, 3 years, more or less. And I'm going to talk about how a piece of my research that is body part based on body part specialization in the colonial hydrozoan, the jellyfish Hydractinia symbioloici carpus, and how this is evidenced through the single cell at the single cell level with gene networks. This is a work that it started in Oxford Brookes University and it moved on into the University of Exeter here in UK. And let me start. But first, so as we know and as we see in the animal kingdom, there's a wide range of forms and shapes. And when we take a look in the phylogeny of all animals, we see that coloniality is present in several clades, in several lineages of metazoans, and it's in also is peculiar because it shows up in distant relatives of the lineages, but more specifically, so it shows up in hemichordates, tunicates, and bryozoans. But coloniality is characterized in the animal kingdom by so a colonial animal is characterized by being modular, consisting of individual clonal zoids that are here they are connected by live tissue. They share a vascular network, and in this vascular network, they have cell types or cells that are migratory, so they move around the whole colony. Another peculiarity is that in all colonial animals, the growth and the reproduction are mostly integrated. A colony works as a single, as an individual. So we call this we classify it as a single physiological unit. So another peculiarity of colonial animals is that they exhibit a robust regenerative capabilities. There are at least in some species, some colonial species, is characterized by having adult pluripotent stem cells. So here we see that it's present in hemichordates and tunicates, so all and bryozoans. So it's all these three lineages that are very distant to each other, but especially in Nigerians. So it's more prominent here, the coloniality, and we see here examples of the Portuguese man of war, the Physalia, the siphonophores. There is all these. It's a colony, a specialized colony, but we also see it in stony corals, soft corals, and sea anemones in the jellyfish clades in the Nigerians. And if we go closer into the Nigerians, we see Hydractinia. Hydractinia symbiologica, well, the genus of Hydractinia is a member of the hydrozoan clade in between Nigerians. In the wild, the colonies start forming when they settle and they grow in the shell of inhabited hermit crabs. So it's not per se in the hermit crab exoskeleton, but in the shell that is inhabited by these crabs. And they attach to the substratum, the colony of Hydractinia, by forming a gastrovascular network of tubes that we're gonna call stolons. And uh, here is the stolonal network where they attach, and they also have a peculiarity that each part of the colony, they have a certain level of specialization. So, or in the polyps, as we say. So, they specialize by the function. So we have the the feeding polyps or the gastrozoids, and we have here the chubby little ones. It's the sexual polyps or the gonozoids. They also have a defensive polyp that is very rare and it only occurs in nature. I don't. It's been documented very few times, and that is called the dactylozoid. It's the defense polyp. Also here, as I have said, as with many colonial animals, the growth relies on a lineage of adult or mature migratory pluripotent stem cells, also known as eye cells, that are just moving around in the whole colony through the vascular stolonal network. More than this, when we see all the resources that are available already for Hydractinia, we see that we have a lot. So we have a genome already. We have several functional experiments with a molecular analysis, like in situ or immunofluorescence, that have been documenting the molecular function of the genes in the colony. We also have an RNA-seq bioinformatic analysis of the colony, but only onto the bulk level. And more recently, we have access now to a chromosome level genome assembly of Hydractinia. So this makes it more appealing for all these studies on studies on coloniality. So from this and all this introduction, we in my research project we had two main questions. So the first one is how different are the parts from Hydractinia in the different colony parts, let's say, and are they made of the same cell types? And also, it's how this interaction of the stolons with the shell inhabited by the hermit crab came to be. Um, yes, and the great advantage right now is that we have a single cell methods, as many have seen in many papers, and these are getting more multiplexable and more low cost throughout the years. So you can now put in the same run of sequencing, you can put different samples, different stages, different individuals, treatments, replicates. You can multiplex it as much as you want, and the cost is going to be similar to a very a high scale experiment that you like a few years ago, like it was unthinkable for any of us to do experiments of this scale. But more than that, I want to explain a little bit how the single cell technologies work. It's basically just a way to tag the mRNAs of a single cell. And we do it through a method developed in the lab here that is called the split-seq, and it's just barcoding uniquely each of the mRNAs of the cells. So we can because it has a very combinatorial power, a very high combinatorial power. We can know specifically where are these mRNAs coming from, in which cell, because we have tagged the cell, we have tagged all the transcripts, and we can group these sequencing results into a matrix that is genes and cells, and we have counts of mRNAs per cell. And after this, we do several. This is where the bioinformatics starts. So here we take all these reads, all these counts, and we process them to generate to perform principal component analysis. We do graph analysis, and we generate what is called a UMAP, which is just a projection of the similarities and differences of the cell types that you are sequencing and that you're getting from your sample. So here we have the atlas as it is of a whole colony of Hydractinia. We got around 200,000 cells that are more or less comprehensive of the whole colony. We managed to annotate 38 clusters and put these into 8 broad categories that I'm later gonna explain a little bit of how we did this classification and annotation. But the way we did it is through several confirmation methods, so we can know what are these clusters that we get. And one of those was the classical. So we did in situs of each of the top marker genes of clusters, the top marker genes of the clusters. And we did in situs of the ones that we think are interesting to us. So we did from several that like here we have the chitinase 2 positive cells. There is a cell type that is specific from glands, and there are digestive and mucosal. We have an immunofluorescence that it consisted of just we developed a specific antibody for this gene, and we label it in the colony, in the polyp, and then we can, it's a specific of the venom cells that are in Hydractinia. But not only this, we because it takes a lot of it's only one way of validating all the clusters. We also used several other methods. So one of them is PAGA, which is just a way of taking all the clusters, all the genes, all the expression data, and compress it, denoise it, and just place it in a multi-dimensional space and perform a graph, basically, where it's just classifying all these clusters based on similarities. Yeah, it's just helping us. This just helps us to establish the relationships between the clusters that we already have. So this helped us to classify most of the cell types that we have into the 8 broad categories that I mentioned before. So we have the epitheliomuscular, we have the epithelial cells, we have cells that are specific from the gametes, all the formation of the gametes. We have there are cell types that are specific from cnidarians. So we have the nematoblasts and the nematocytes, which are these stinging cells that are triggered through mechanical input. We have the neurons, we have the gland cells that I mentioned are it could be either digestive as well, and we have the progenitor cells that are that they are grouped on their own, because it's already, as I mentioned before, they're already matured, these progenitors, these stem cells in hydractinia. But not only this, we took the expression data and we generated a co-occurrence of the whole set of all the cell types. So we took the 85% of the highly variable genes that have a fold change bigger than 1.5. So all this is technical, but it's important to understanding the why we generated these, why we reached to the conclusion of the relationship of these clusters. And so we took the highly variable genes, we did a hierarchical clustering of these cell types, and we generated a Pearson correlation of the separation of these two, and we did the Pearson correlation between these two iterations of the data. And we generated a co-occurrence matrix that is just telling us basically what we more or less knew already, that is the epithelial and epitheliomuscular are two broad categories that are very related to each other. They have a lot of expression ways similar to each other. We see also that the neuronal cell types and the nematocytes, which is all these stinging cells from the Nigerians, they are related, not as tight as the epithelial and the epitheliomuscular, but they have some relationship, some tight relationship between each other. And we see that the progenitors, so the stem cells, the progenitors of the gametogenesis, they are on their own here, basically. So all this it helped us classify and validate all these cell types into these 38 clusters, and it helped us to resolve the major cell types in the colony. So as I mentioned, the neurons and the nematoblasts, and it also helped us to annotate novel cell types like, and I'm going to talk a little bit later, the pre-Silkin positive cells. And so now we have, with single cell transcriptomics, we can pinpoint all the parts of the colony in the UMAP in single cell atlas, and we can now, just by seeing at it, that there is an unequal distribution of the colony types in the single cell atlas. So we can take this information and compare all the different parts of the colony and calculate ratios and do statistical tests on it. And so we did. So we used a paper, a software developed to do a compositional analysis based on Markov chains and Bayesian modeling that is called SCODA. And we generated these bar plots with different levels of significance. But it's worth highlighting here that when we compare the different parts of the colony, the polyps in general are enriched when compared to other parts. They're enriched with epithelial and epitheliomuscular cell types. We see that the sexual polyps are this is a bit obvious, but we needed to know statistically that the sexual polyps are enriched with germ cells and mixed progenitors. All these cell types are responsible of gametogenesis. And we see that in the feeding polyps, they're enriched with cell gland clusters with nematocytes and conolipin positive cells, which are which are these venom cells that are they haven't been that much studied in hydractinia, at least. But more importantly, we see in the stolon, so the cell type that is more in contact with the substratum, we see that it has fewer neuronal and fewer gland cells, but it's abundant in pre-silkin positive cells, in nematocytes and conolipin positive cells. So this is already telling us um, like it's helping us to answer our first research questions are these what are the different parts of the colony made of, and we can successfully say that the colony parts seem to be defined by a combination of cell types and by different proportion of the shared cell types. So it seems to be doing a mix and match. So it's taking what is available at the moment and then build up from that. The colony part that wants to build. But not happy with this, we decided to go further into the bioinformatic analysis and to study more what are the molecular the gene relationships of these cell types. So we decided to implement an R package that is called WGCNA. It's been published for a while now. It's going to be 20 years. That's how I know I'm old. And it just takes a different correlation between the genes of a certain data set, measures co-expression, and then classifies that into different modules of co-expression, so to say. And you can use this for the comparisons that you want. So you can overlap this with gene ontology terms and just take all these these gene sets, gene modules, and then compare it as much as as your imagination allows it, basically. So we did this, and it helped us to, when we overlap it with the single cell data set of hydractinia, it helped us depict a regulatory landscape of cell differentiation in the whole colony. So in here in the left, each dot it represents a gene, and the edges are a representation of co-expression scores. So it tells us the similarities and the relationships of the different cell types in the genes at the gene level. And on the right is just a different representation of the network, of the gene regulatory network of the whole colony. That is just a summarization of all the genes in a cell type, and it's just each dot. It's a cell type, and the edges in between the nodes is the summarized co-expression. It's just showing us a bit more clean the associations between the cell types. But yeah, this is a pretty visualization. But if we take the network as it is and we see how does it correlate, how does it, well, not correlate, overlap with the cell clusters that we have, we see that each module or most of the modules are specific of cell clusters. So it helped us to characterize the gene ontology terms of each gene module and each cell cluster. And as an example, and here it helps us to depict a regulatory landscape already of the whole colony and all the associated biological processes in the annotated clusters. And yeah, as an example, we see here very specifically genes that are in the pre-silkin positive gene module that are also in the cell in the specific of the pre-silkin positive cells, and it's yeah, but I'm gonna talk a little bit of this later on. When we take this same network and we overlap it with information of transcription factors of databases that are already very well annotated, we can take information to nurture the gene regulatory network. And it's just here is taking all the top central transcription factors of each gene module and in which cell type do they belong to. And what does it mean? What do I mean with the top centrality? It's just that it's just telling us the factors from these, all the transcription factors in a network, in a specific cell type, in a gene module, which is the better connected. So which one has the more number of connections in the network. So it's and it's just a good measure of saying, of identifying regulators of a specific cell type. So yeah, when we do the same as previously, and we do the heat map with only transcription factors, we see that they're very specific, either to broad categories or to cell types. And yeah, it just helps us to identify candidate regulators of each gene module and ergo each cell type. And as an example, we see here a very specific transcription factor called HES2, and it is very localized into the pre-silkin positive cells. But yeah, now we wanted to study more this cell type and this gene module, because when we see the geo terms, the gene ontology terms, the biological process associated with both of these are seem to be biomineralizing processes. So chitin metabolism, we have tissue development, we have skeletal muscle cell differentiation. So we aim further in this point of the research of the paper to investigate the nature of the pre-silkin positive cells. So, however, when we take some of the genes of this gene module and this gene cell type, sorry, when we try to do a simple blast, it seemed almost surreal because it doesn't throw any significant match to any of the databases in NCBI. So where do we go from here? But yeah, but first, when we look at the genes that weren't giving any match to this, to any known annotated gene in NCBI, we see that there have been previously described as a crucial part of the biomineralization matrix, typically in mollusks. So we see here the pre-silkin 39 and the schematrin, that is like a family of proteins that are very rich on glycine repeats. And even more than this, when we take the cell types of these the genes that are in these cell types, so the pre-silkin positive cells, and we do a reciprocal blast with an already published single cell atlas of a coral, you would think that the pre-silkin positive cells will be having a high score of similarity with the cell type in Stylophora piscillata. That's the other color. That is the other cell type that is responsible of developing the calcareous exoskeleton in Stylophora. But no, it seems like it has minimum relationship, so it's not at all similar transcriptionally speaking to the calicoblast in Stylophora when we do a one-to-one ortholog scored expression from the hydractinia genes in the pre-silking positive cells. Um, we took we managed to annotate some of these genes, and we saw that some of these are a part of the biomineralization pathway. So like chitin synthase, protocadherin, alpha carbonic anhydrase. And when we do a gene tree of each of these, we see that they're like it's verified that first that they are these genes indeed, and that they are unique. No, not unique. They are a Nigerian characteristic. Um, but further, when we see the organization of the pre-silking genes and the shematrin genes, it is they are very close to each other in the same section of the chromosome. And but when we so we develop a custom script that is just going a bit beyond, since we couldn't find any any ortholog similarity through blasts. We develop our own script that is just trying to identify these glycine repeats in hydractinia. And so we did. So it's like a visualization of the patterns of the glycine repeats in between flanking amino acids. And so when we were wondering if if the pattern of glycine repeats is the same on the rest of the cnidarians that are annotated in NCBI, and we see that they have somehow similarities in the peptides, in the signal peptides, and in the glycine repetitions, but not as much as when we apply the same script in mollusks and brachiopods, which are other like a bunch of other biomineralizing species. So what do we get from here? Like is like that the same repeats are found that are found in the shematrin, and the shematrin in hydractinia are more similar to mollusks and brachiopods than any other cnidarian even from the same clade. And more than that, when we isolated the gene, the pre-silkin 39 gene, and we did fluorescent in situ hybridization. And we see that it's indeed localized. So this is a forming colony from below. So this is the polyp, and this is the beginning of the stolon network. So it seems like the pre-silking is like right at the beginning of the interaction of the stolon with the substrate. So um uh, so yeah, it's the pre-silking is here, and it's more similar to mollusks than to other cnidarians indeed. So what can we get from this to help to answer the second question that we had? So how is this interaction of stolons and the hermit crab evolve? So we see that the pre-silking positive cells in the stolons are capable of generating a biomineralization matrix that is very similar to mollusks. In addition to this, the stolons are highly enriched with allorecognition positive cells and other cells that are just the as I told you, the mix and match organization of the different parts of the colony. But more importantly, is this capability of generating a biomineralization and an allorecognition that is like resembles the shell matrix of a mollusks. So as a summary, we can say that the single cell landscape of the whole colony, it just uncovers the first steps, the principles of the gene regulation across all the cell types in the colony. We see that the different cell type proportions of the different parts, they seem to be using this mix and match approach, where it's just as I said before, it's just taking all the cell types that are available at the moment and build up from that the different parts. The additional to this, the single cell atlas just helped us to annotate newly Nigerian cell types, which is in this case the pre-silicon positive cells, the allorecognition cell types, and the stolons that are enriched in the stolons. And when we take a look at the stolon cell type, we see that the biomineralization factors are, first of all, specific of stolons, but when we compare it to other biomineralizing species, it shares more similarities to mollusks than to other cnidarians even. So yeah, just to finish here, just found out on this Monday that our paper has been published in Nature Communications, and yeah, it's a huge achievement. And with this, I want to thank all the collaborators in Galway, in New Zealand, in Bristol, in Spain, in all the people that help into building this a beautiful story.
Wow. I mean, I was here, looking slide after slide and just thinking, what a wealth of knowledge. Really. No, 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 no. It's not a surprise that you have so there listed so many collaborators. So my understanding is that this model species, well, this species, let's say, has become a model species because of the collaboration of these groups. Is that right? Over the years, these groups have been working with these species, and then now you guys have built up the molecular toolkit and molecular knowledge to use it as a model species for coloniality. What do you
The
think?
yeah the colony has been maintained in Galway in Ireland by Uri Frank, the lab of Uri Frank, and they basically wanted to know all what I'm telling you, because they already had RNA bulk sec, and they just wanted to know at the single cell level, and they just went to my boss and started asking, like, can we do this at the single cell level? And they, like, more people started getting involved, and it's like at the end it helped a lot to build this amount of results that we have right now. So, but yeah, we here in Exeter, we don't have the we don't work with the animals directly.
Fair enough. Well, but that's a great example of how, international collaborations, how far they can go, right? So this is great. I think
That's true.
science is inherently collaborative, so it's great to
Mhm.
see that. Now, I do have a few questions, and like I told you before we started recording, my questions are going to be basic, right?
Yeah,
Because I'm a
no
little far
problem.
away from your field. But again, thank you very much for the talk. This was really, really interesting. And also, congratulations on the Nature paper. Great timing, right? The paper just came out.
Yeah.
We're recording this today, so congratulations. Good timing. Now, my first question is, you've shown in a few slides, I've seen the eye cells, but I think you haven't mentioned what they are. What are they, if you haven't mentioned? If you
Sorry, maybe I missed
go
it,
back,
but yeah,
if you go back to, I
the
think, like, two or three slides, one of the final slides just before the summary, there was Right there, global. You have
Yeah.
eye cells, epithelial. What are the eye cells?
The eye cells are basically stem cell-like cell
Mhm,
types
mhm,
in cnidarians. So they have a lot of features that are common of stem cells in mammalians,
mhm,
but because the field doesn't want to classify them as stem cells
mhm,
yet, at least as
mhm,
far as my knowledge goes. They just call them eye cells.
mhm. Yeah, I was just curious because the
Yeah,
other ones, I mean, you can tell by the name, right? But
yeah.
those ones, I was thinking, what are they? Um, I thought somehow I was thinking of interstitial, like, the cells that are in the middle of the tissue or something like this. So yeah, I was just curious. Now, so you've shown us a very detailed body of work in terms of dissecting the molecular signatures of coloniality in this particular model system, right?
Mhm.
Um, and then in one of the answers to the first research question you're showing there right now actually is that colony parts are seem to be made of this mix and match strategy in which, like you mentioned a few times, the colony there will make do with whatever it has, right?
Yeah.
I was wondering whether you would know or whether, people are doing this. Perhaps they aren't yet. What about the Evo Devo behind it in terms of, I guess, embryology or histology even? Because I'm just wondering about the biology. So let's say you grab a stolon or other piece of the colony, which has a varied cellular composition. Allegedly, let's say allegedly, each and every little piece of the colony will have all the cells, all the cell types that are needed to build a new colony.
Mhm.
But yeah, how does that part of the colony decides, oh, now let's grow, I don't remember the names of the parts of the colony, but you know what I'm saying? Like what determines, because
Yeah.
each colony part, although it has a varied cellular composition, there will be a signature, right? So
Yeah.
some colonist parts will have more cell type, one cell type than the other. I was just curious as well. Have you seen are you guys doing this, or have you seen this in the literature?
So there are, to my knowledge, there are several factors influencing there. So the first is that the Hydractinia and Cnidarians, jellyfish in general, they seem to be very plastic, more plastic than any other animal, like similar to sponges, where you can just take a chunk and then you can build. It grows back the whole organism as with a few couple of cell types. Um, so it seems to be not an issue when a part of the colony is missing,
Mm,
because they have already everything, all the machinery already is there,
mm,
present in all the parts of the colony, so they can just start from scratch and they don't seem to care. The other thing is that there is at the beginning of the colony, it seems to be a molecular, a morphogen, not a morphogen, a molecular gradient of a gene expression. So it's similar to an organizer, like an anterior posterior organizer in other organisms, and it seems to be working with Wind A, I think. Maybe my collaborators in Galway will kill me for this, but I think it's a Wind A,
mm,
a gene that is just generating the gradient that is signaling the what it is in the colony, that this part is where is the gradient is it's different. This is the stolon or this is the polyp, and then from that, just like the cell division just creates the gradient and then starts differentiating from there. There is I think it came out last year, a paper on reaggregation, cell reaggregation in hydractinia, that are our collaborators in Galway. They are working extensively into that to know what are the factors influencing into like, for example, you dissociate a whole
Mhm.
colony and then how does it reorganize? Does it reorganize? How does it do it? Like all these kind of questions are, I think it came out also this year or late last year, but yeah, they can tell you more in detail of that.
No, sure, absolutely. And as you're explaining this, I was even thinking perhaps even physical attributes of the environment might
Yeah,
actually trigger certain, molecular responses, right? So whether, for instance, the specimen, is stationary, whether there is a particular, like water current, temperature, salinity,
yeah,
I think, yeah, those would be interesting factors to look at if they haven't been looked at already.
yeah, exactly.
Now, one last question. Uh, so you've told us, you've shown us that coloniality in metazoans has evolved independently in distantly related species groups, which I
Hmm.
think is fascinating, right? It's
Yeah.
a very nice example of convergence, and I think we would not run short of ideas of how adaptive coloniality can be, right? Uh, but I was wondering whether, and again, this is speculation, whether you would expect a a common denominator, let's put it this way, a common denominator across those groups, and I want to say a molecular common denominator across those groups, that it would allow or would equip those groups to develop coloniality, right? Because again, they are all metazoans, they are highly
Hmm.
differentiated, they had, different cell types, but we don't see coloniality in all groups, right? So
Yeah.
one must wonder, do they share something common? Or alternatively, all other groups that haven't evolved coloniality, do they have a barrier to coloniality, right? Again, I'm just speculating. It's really thought-provoking.
Hmm.
Have you thought of it? What's the common denominator? What's the molecular signature of that trait? I mean, you've shown us it's complicated, right?
Yeah.
The
It's complicated because if you like you also have to think about what do you define, where is the line where you define coloniality and let's say multicellularity,
Mm, mhm,
because in that case sponges are also colonial in
mhm,
some sort of way of speaking,
mhm,
but I think coloniality factors may be enhanced by features on the genome, perhaps,
mm,
that are just allowing for the different parts of a specific colony to specialize, so to
mhm,
say. As is the case of hydractinia, that one polyp becomes sexual, one polyp becomes feeding, and then we have all the network that is to attach ourselves. So yeah, maybe it's in the genome, maybe because not even you may think that the ecology of these animals is influencing somehow, but not even because they like, for example, bryozoans live mostly in fresh water, and in brackish water or high current water. So it's not the siphonophores, they just wander off the ocean the whole time. So I
They all seem to
wouldn't.
be relative.
Yeah,
I'm thinking about the ecology, but again, it's a chicken and egg problem, right? Because I was gonna say, oh, they're at least they're, if you will, their trophic mode, right? They're all kind of grazers, or they're just, filtering, right? They're just filtering things in the environment. But again,
mhm.
that might be just actually what they can do once they're colonial, right? Because if you're a colonial,
Yeah,
you're not going to go and hunt things because you're not going
yeah,
to have your colony around with you, right?
yeah,
It's almost like they're limited, because as we know, right? So for each and every body plan, for each and every um, life history strategy, there are constraints, there are advantages, but also disadvantages. But yeah, I think, I don't know, I must say, I was looking at your slides and listening to your story, to your results here, and I kept thinking, I was like, oh my goodness, this is very interesting, and I kept thinking about what the other groups would look like at this level. I think eventually,
yeah.
one thing that perhaps you in the future or your group or collaborators could look at, if people aren't already looking at it, is compare with coloniality in, let's say, simpler groups, right? Because we know, for instance, in, again, I guess, I mean, fungi, I would say they must be considered colonial, and
Yeah.
some green algae as well. I would say, again, I don't want to say anything wrong, but that would be my bet. In my understanding, is that for simpler, and again, simpler between huge quotation marks here, for simpler groups, I think um, becoming colonial is, well, perhaps I'm mixing up with becoming multicellular, because I know, for instance,
Yeah.
in green
Mhm.
algae, the evolution of multicellularity is mostly explained by the fact that, the particular species has failed to hatch, right? So you reproduce, you create your
Mhm.
offspring, but that offspring, all those offspring, they are they carry on stuck to each other. They
Yeah.
haven't hatched and become independent. So that might be just multicellularity, not coloniality. But anyways, I'm
Yeah.
again, I think I know very little, if anything, about this, but I think it's fascinating, and I think it's certainly
Yeah.
worth thinking
No,
about.
and if I may add, like an important factor when you're becoming colonial is this here, the allorecognition between the different individuals. And that is something that so coloniality sometimes the study of coloniality sometimes gets overlapped with a study of the evolution of the immune system. Uh, so yeah, there is something there.
Because you need to recognize self versus non-self, right? You need to accept the different types of self, and also say, "Oh, they are all part of my colony. Let's not
Yeah,
attack." Um, Anyways, well, David, I think that's enough of questions. I've already interrogated you long enough. Thanks again for taking the time to speak with me. Thank you again for bringing this wealth of knowledge to all of us today. I really appreciate it, and I wish you a best of luck in your future steps. And
perfect.
I'll be watching. I'll be looking forward to seeing your upcoming publications. Okay?
Thank you very much for the invitation, Matthews.
Thank you.